This is the recording for the slides for Chapter 12 on the cell cycle. And mostly what we're focusing on in Chapter 12 is a process called mitosis. So what we are seeing in slide number 2 is that multicellular organisms start out as a single cell that divides and produces 2 cells, and then those cells divide and produce 4, and so on. So that's what you're seeing in this image here, is where with mitosis we're mainly talking about learning about how multicellular organisms grow. They begin as one cell, which we call the parent cell, and in multicellular organisms such as humans, us, we begin as one cell called a zygote, and that zygote formed from when our father's sperm fertilized our mother's egg. And so the first cell that began dividing into what would become us, that first cell is called a zygote. So as that first cell divides, when it divides into 2, that process is mitosis. When those 2 divide and divide and divide, until you have the trillions of cells that form the human body, that process of division is called mitosis. The phase of cell division that occurs when the cell is not actively dividing is called interphase. And during interphase, the cell will grow in size, it will get larger, and it will prepare to divide again. It will prepare for cell division, unless it is a cell that has stopped dividing, that will no longer divide. For example, if it's a mature nerve cell or neuron, then it won't divide again. So it will remain in interphase, basically, in a stage called G0. But it will no longer prepare for division because it won't ever divide again. There's a few cells in the human body like that, like our nerve cells or neurons, and our some of our muscle cells are like that. They won't ever divide again. But most of our cells are able to divide again, and they divide by a process called mitosis. During mitosis, the chromosome copies that were produced during interphase, and you can see here this parent cell has one single-stranded chromosome. During interphase, that single-stranded chromosome became a double-stranded chromosome consisting of 2 sister chromatids, and those sister chromatids are identical. They are absolutely 100%. The genetic information is exactly the same. So they are identical to one another. So during interphase, all of our 46 chromosomes will replicate, and they will each contain 2 sister chromatids that are genetically identical, so that during mitosis, when those sister chromatids separate, each new cell that forms, is called a daughter cell, and each new cell that forms will have an exact copy of our genetic code. And then after, or actually at the end of mitosis, in a stage called telophase, during that last stage of mitosis called telophase, there's another process that occurs called cytokinesis, and that is when the 2 daughter cells split in half. And they're going to be clones of the parent cell. They're going to be genetically identical to the parent cell. There's a video on sea urchin division, cell division. This is slide number 4, so I encourage you to watch that video. And then the reproduction of cells is also known as cell division, and that is what this chapter is about, chapter 12. The key roles of cell division are, single-celled organisms can divide and produce new organisms by asexual reproduction. So if a single-celled organism divides and produces two new organisms, then that is asexual reproduction. That original organism has cloned itself. But multicellular eukaryotic organisms like us, anything in kingdom Protista, kingdom Plantae, kingdom Fungi, or kingdom Animalia, are all eukaryotic. So anytime you have multicellular eukaryotes, when the embryo is developing, the cells are dividing. More and more cells are dividing and being formed. So that is one role of cell division. And then cell division then plays a role in renewing and repairing cells in multicellular eukaryotes that are grown, that are mature. So they still have to use cell division to repair themselves. Or like, for example, you probably know humans shed skin cells. Our outer layer of skin is shed around every 3 weeks. And then we replace that layer by, we have a layer of skin cells that undergoes mitosis and produces new skin cells to replace what we lost. So replacement, repair, that's the role of cell division once we become adults. Not so much growth, but replacement and repair. So you do need to know that all the DNA, in other words, all of the genes in our cells, for one thing, every cell that we have, every cell in the human body has a copy of all of our genes. And all of our genes together is called our genome. So every single one of our cells, no matter what type of cell it is, has a copy of our genome. Just because it has a copy, it doesn't mean all those genes are turned on, but it does mean that our skin cells, our hair cells, our muscle cells, our bone cells, every cell in our body has a copy of all of our DNA, every single bit of it. Not just the genes that are important to that cell, but all of the genes, okay? So our DNA inside of our cells, our DNA that contains every single one of our genes, is packaged along with proteins into structures called chromosomes. And every chromosome can carry up to a few thousand genes. So in humans, we have 46 total chromosomes. 23 came from our mother and 23 from our father. Now, when our cells are not actively dividing, when they are we won't say at rest because they aren't necessarily resting either, but when they're in the stage that we call interphase, the chromosomes are not condensed into structures that we can see and count. Chromosomes usually look like either a single strand or they look like the letter X, because those sister chromatids are attached at one spot, and so they actually appear like the letter X in the alphabet. So when we can see the chromosomes and count them, that is during mitosis. But when interphase is going on, when the cell is not actively dividing, those chromosomes decondense, I guess you could say. They spread back out, and you can't count them anymore. They're so diffuse and spread out that the DNA is called chromatin, okay? So when the cell is not actively dividing, the DNA exists in a form called chromatin, which is very spread out. And then when it is actively dividing, that chromatin condenses and forms chromosomes, which we can see and count. So every species has a certain number of chromosomes in the nucleus of their cells, and every species has two types of cells, basically. Somatic cells are body cells, and gametes would be egg or sperm. Gametes would be sex cells. So all of our cells that we call somatic cells, all of our cells besides our sperm or egg, will have 46 chromosomes, or 2 sets of chromosomes. And then if we are male and we produce sperm, that's called our gametes. Our gametes are only going to have half the number of chromosomes as our somatic cells, because we only give half of our chromosomes to our offspring. So our reproductive cells, or gametes, will only have half of the chromosomes. So in humans, sperm and egg will only have 23 chromosomes, whereas all the rest of our cells will have 46. Now, before a cell divides, DNA replicates or copies itself, and that DNA, that chromosome that was just a single chromosome, duplicates, and now it consists of 2 sister chromatids. It's attached by proteins called cohesins at an area that we call the centromere, which kind of looks like a waist. It's thinner than the rest, I guess you could say. These are 2 sister chromatids joined at the centromere, and so they kind of look like, under the microscope, like the letter X. So once the cell starts dividing, those 2 sister chromatids separate, and each daughter cell gets one of the sister chromatids. So this is kind of what it looks like in an illustration. When you start out with a parent cell, the parent cell has 46, if this were a human parent cell, it would have 46 chromosomes that are single-stranded. And then during interphase, those chromosomes would replicate, and each chromosome would have a copy. So they are now called sister chromatids. They're attached at a region called the centromere. And then during mitosis, those sister chromatids separate so that each daughter cell gets a copy. Here's an animation on mitosis. This is slide number 16. I would highly encourage you to watch this animation. And what you're looking at here, by the way, in the animation, you are looking at a stage called anaphase, A-N-A, anaphase. Anaphase of mitosis is where the sister chromatids are separated and begin moving to opposite ends of the cell. All right, so some definitions. Mitosis is defined as the division of the genetic material in the nucleus, and cytokinesis is the division of the cytoplasm. Cell division involves both mitosis, which is division of the nucleus, and cytokinesis, which is division of the cytoplasm, or the cell itself actually splits. Now, gametes are produced by meiosis, which is chapter 14. And meiosis does not produce genetic clones of the parent cell. Meiosis produces daughter cells that are not identical to the parent. All right. The cell cycle consists of the M phase and interphase. The M phase is the mitotic phase, which includes mitosis and cytokinesis. So the M phase is the phase where the cell is actively dividing, and then interphase is the phase where the cell is not actively dividing, but it's not resting either, because a lot of things happen. The cell grows, it replicates or copies its chromosomes. In other words, it produces those sister chromatids, and it prepares for cell division. It produces proteins that are needed for cell division. So let's talk about interphase. If you look at the life cycle of a cell, it spends about 90% of its life in interphase, in the process of interphase. 10% is spent in mitosis, or the M phase. So interphase consists of G1, S, and G2 phases. The S phase is when the DNA replicates, because think of S as synthesis. But during the G1 phase, there's metabolic activity that's consists of such as cellular respiration and cell growth. One thing that happens is every one of the cells are going to have organelles like mitochondria, lysosomes, Golgi apparatus. So every one of those organelles that's found in the parent cell has to copy itself. So the daughter cells will have a copy as well. So one thing that happens in G1 is that the organelles inside of the parent cell double. Then in S, the DNA replicates. The sister chromatids are produced. The chromosomes are now duplicated chromosomes with 2 sister chromatids. Then in the G2 phase, you have more cell growth and proteins like proteins that we call spindle fibers, which are a type of microtubules. So microtubules are produced that are going to be part of the spindle that we're going to see during mitosis. And so these are proteins, these microtubules or spindle fibers are proteins that are going to be important in cell division. So that is what happens in G2. Now this is slide number 21. We're going to stop here and I'll start the next recording at slide 22.